Hello, everybody. Thank you so much for joining us. I'm just going to give it a few more seconds for everybody to get on the line here, and then we will go ahead and get started. All right. Thank you so much for joining us today for our third annual virtual event of Connecting, Learning, and Healing. On behalf of my co-conspirators, Joanne Schill and Monica White, well, along with the Colorado Tick-Borne Disease Awareness Association and the Colorado Lyme and TBD Support Group, we thank you for joining us today. Wanted to just take a quick moment to thank our sponsors, because that would not happen without them, IGenix and Insect Shields. Insect Shields does have 2 door prizes, which I will be IMing the 2 lucky winners at the end of the program today. So you must be present to join. If for whatever reason you aren't able to receive my message, I will go on to the next individual. Since we do not have a lot of time today, I will be doing it that way. As far as Colorado Tick-Borne Disease Awareness Association goes, I just want to call out that our focus is on education, prevention, research, advocacy, and patient support. And we did get our proclamation from the governor of Colorado again this year. We have since 2017, stating that May is Lyme Awareness Month. Also just want to call your attention to the corner of the screen there of the multiple work groups that Monica is working on. And we wanted to really highlight this because, you know, this is Lyme Awareness Month and all of us together make the difference. We talk about it, we share about it, and we move that needle to the point where they can no longer ignore us or our disease. And 2 gentlemen who have been phenomenal advocates for all of us, Dr. Burrascano and Dr. Kindleiner, are here today to present. So thank you both for being here. Couple housekeeping items before I introduce the gentlemen. First up, Dr. Burrascano is going to be going first. He's going to be speaking with us on Bartonella, diagnosing and treating the stealth infection. He will go for 1 hour, and then Dr. Kindleiner will join us, and he will be discussing with us a toolkit for promoting your own healing. There will be a small Q&A at the end of each presentation, but because we are limited on time, please limit your questions to 1. And this is being recorded. I will get this posted next week when my tech support person is back in the country, and we'll get that up on YouTube and out to everyone. We won't be taking any breaks, so please take care of yourself. One last thing, I just wanted to show, we have our website, our Facebook, our Twitter, our Instagram, also the Facebook page for the Colorado Lyme TB support group. That has been in the past a private, it is still private, but we had it pretty locked down. You can now find us if you would like to join us. But please be mindful, Joanne will ask you how you heard about us. And we're doing that to make sure that this group of 600 strong stays safe from trolls and solicitors. Okay, enough of that. Let's get to why you're all here. Dr. Burrascano, he is going to be speaking with us today about Bartonella, diagnosing and treating the stealth infection. Dr. Joe Burrascano is a well-recognized specialist in the diagnosis and treatment of Lyme disease and associated complex infection diseases and the chronic illnesses that accompany them. With over 35 years of research in the research field, he has appeared in and on virtually every form of media, both in the United States and abroad. He has testified again with the U.S. Senate and Armed Service Joint Subcommittee and at various governor's consulates. He is also the founding member of ILADS and ILADS Educational Foundation. Dr. Burrascano, thank you so much for your ongoing support. I'm going to hand this over to you.
All right. So thank you for that. Nice introduction. And hello, everyone. Thank you for joining us today. You can see I'm wearing my Hawaiian shirt here in New York. It's nice and sunny and hot. I thought it just makes the mood feel better. So, you know, the organizers gave me permission to use give any topic I wanted. And I've talked a lot about Lyme disease. I've talked about testing. I've talked about other things. But I want to spend the entire time on Bartonella, because Bartonella is such a difficult illness for our patients and for the practitioners. Many consider it a stealth infection because it's very hard to detect and hard to even diagnose, even think about. So I'm getting paged. All right. It's a reminder to not forget to do the talk. See that? I'm only a few minutes behind. All right, let's just get started. Bartonella. All right, what's the big deal about Bartonella? First thing is that there are at least 40 different species that are recognized. A year ago, I gave this presentation on Bartonella, and we talked about 30 species recognized. So you see over time, more and more are coming to light. And not only are there many different species, a patient can have more than one at a time, can be co-infected with the co-infection. So that makes it much more difficult. It's a parasite that can live inside the cells, but can be found outside the cells as well. It can be in the blood circulating, can also be in the tissues, can even be deep in tissues, such as in bone. Not only is it found in mammals, but also birds. So it can be transmitted and kept in the environment, in many different ways. One of the biggest problems with Bartonella, more than any of the other co-infections, even Lyme itself, is the fact that this is a very robust builder of biofilm. When biofilms are formed by an organism, it adds to the chronicity of the disease. It makes it difficult for the body to get rid of it. It inhibits immune response. Also makes testing and treatment very difficult because things that you want to get into the biofilm to find the Bartonella can't do it. That includes antibiotics as well as testing things, which we'll talk about. Bartonella is also very common in Lyme patients. So it always has to be considered, always, even if someone has pretty clear-cut Lyme symptoms, you have to think in the back of your mind, is Bartonella present? And the reason why is because there's so many different ways people can get Bartonella. Obviously, ticks are an important vector. There's some controversy on that, and I'll show you a slide that hopefully disproves that. But also it's known to be in fleas, mosquitoes, biting flies, even mites and red ants. I saw a recent article on the red ants. Obviously also animal bites and scratches. That's why one of the versions is called cat scratch disease. And in the medical field, veterinary field, in cases due to needle sticks. It's also well known that it can be a maternal fetal transmission. So hard to avoid this thing. The distribution is also worldwide. It's not even regional, it's everywhere. Different regions might have different strains or species, but it's even been found, believe it or not, above the Arctic Circle. And again, the basic idea is that it's very difficult to diagnose, very difficult to treat. So here's a picture. This is an article, Fleas and Arctic Foxes from Nunavut, Canada. I put the picture of the globe up, and this is where it is, way, way, way up north, at least as far north as northern Alaska. So if it can go there, it can certainly become in our backyards. And, obviously it's not just the Arctic, it's been found in pretty much every state down here. As far south as Hawaii, Florida, as far as northern Alaska and northern Canada. And there are many different species. The more common one is called cat scratch disease. It may not be the most common infection, but the most common name for it. Trench fever is another one that's known well, especially in Europe. So here's about the tick bite. Two different articles. One is a person who had a tick bite, developed a rash, developed regional lymphadenopathy of swollen lymph glands, and they proved that it was from Bartonella. Second one is a study where they actually did transmit Bartonella from a tick to an animal, in an experimental setting. So, his proof here and published that it can be transmitted by ticks to people as well as to animals. Here's another thing that I was thinking about. This is not from an article, but there was one report that they found Bartonella in the salivary glands of the tick. When ticks bite someone, the first thing they do, as soon as they pierce the skin, is they inject their saliva, because the saliva has agents that numb the skin so you don't feel the bite, has agents that inhibit the immune system to allow the tick to not get attacked from your body's immune system, and therefore also the germs that they harbor. But if there's therefore something in the salivary glands and in the saliva of the tick, that'll get into the person as soon as the tick really bites into you. Now we know that certain viruses are kept in the saliva of ticks, and they can be transmitted as soon as a tick bites. We also know that about some of the relapsing fevers. So the fact that Bartonella has been found in the salivary glands of a tick means that it's very likely, and it's no proof to this in terms of articles, but it's very likely that you don't have to have the ticket attached for 12 hours, 24 hours, 36 hours. It can be like right away. All right, now, why is it so hard to diagnose? First of all, most symptoms are not specific, and traditional testing is notoriously insensitive. In the lab or in the office, I was going to say, any one test, we're lucky if we pick up a third of the cases or 20% of the cases. So what we end up having to do, and I'll get through this, is to do a bunch of different tests of different types to try and find it. It's well known as a cause of what's called FUO, a fever of unknown origin, because again, testing is so poor. Someone comes in with a low-grade fever, a chronic disease. They go through every test and every culture known to science and nothing shows up. Then you have to think fever of unknown origin. In fact, it's a very favorite question on medical boards, a medical exam, to say, what's the cause of fever of unknown origin? I have the slide as well. It says brilliant. Bartonella endocarditis is often missed. The Bartonella can actually infect the lining of the valves of the heart, the parts where they come together and they get kind of eroded. The surface is a little bit raw. It's natural that way. But the Bartonella that's in the blood with their biofilm can get into that raw surface and start an infection. And the problem is these Bartonella can break off and seed the entire body with a constant shower of germs, but the core of this is on the edge of the valves. And that's where it starts. And the problem is that with this bad testing, you would not know what's going on. And the only way that these are often found is that someone ends up with a destroyed heart valve. They have surgery to put an artificial valve in. And after the fact, they look in the valve and say, oh, wow, it's Bartonella in there. Well, for that reason, it can be isolated from the blood, spinal fluid even, from the GI tract, including the appendix. I wonder how many cases of appendicitis came from Bartonella. It can be found in the liver, in the skin, in the bone itself. There are very many reports and actual photographs where you see Bartonella actually in the hard part of the bone adjacent to the joint. It can be found in the cartilage too. And cartilage is a part of the body that doesn't have any blood supply to it. You've seen cartilage when you've like had chicken and meat and so forth. There's no blood supply there. There's a hard rubbery substance. If the germs can penetrate that without a blood supply, then how are you going to get your immune factors and maybe antibiotics into that area? Very hard to do. They're found in lymph nodes, and very often someone will present with swollen glands. And I had a patient who was 1 week away of getting anti-cancer treatment for what they thought was a lymphoma. And we found it was Bartonella, treated him appropriately. The lymph glands went away. The diagnosis was changed and saved him a wrong diagnosis and toxic chemotherapy. It's been found in stem cells. It's been found in heart valves, as I said. I mean, it just goes everywhere. The other thing that's been known, and there's a reference there, is that it can go from mother to child, not just humans, but animals as well. Another thing that is very interesting, I find, is that Bartonella, when it's present, can also activate autoimmunity. So now many times you say, well, for example, with Lyme disease, it gets in the joints and mimics autoimmunity. Well, this can do that too, but this actually can cause autoimmunity itself, lupus-like syndromes, inflammatory bowel disease, and so forth. And when you get rid of the Bartonella, if you can do it, the autoimmunity goes away. So it's a very interesting aspect of this, because it's more than just an infection in the joint. It's where your immune system not only can't get rid of the infection, but actually starts to work against the body in a very active way. And along the same lines, there's an enzyme or a chemical in our body. It's a vascular endothelial growth factor. It's called VEGF. And that's responsible for the growth of new blood vessels. A number of tumors, especially breast cancers, secrete this VEGF, and they cause more blood vessels to grow. It's a way for the cancers to spread and become a problem. In breast cancer, there's a kind called inflammatory breast cancer that has the spread of the tumor through all the lymphatics and the blood vessels very widely. It almost looks like an infection. It's like red and angry and inflamed. And it's been found when you biopsy these people, guess what's present? Bartonella. Now, does Bartonella cause breast cancer? Probably not. But if Bartonella is present in the setting of cancer, especially breast, or one that's known to stimulate the VEGF pathway, it's going to probably make it worse, more likely to spread, and be more difficult to treat. So as I say, it's hard to diagnose, but there are some clues. One of the main findings, clinicians will tell you this and patients too, there are symptoms involving the central nervous system that are out of proportion to the physical ones. So for example, if you have a Lyme patient who has brain fog and not, thinking clearly, and maybe some irritability, trouble sleeping, but they have terrible joints and they're very tired and nerves are tingly and so forth, that's kind of all proportionate. But if you have a patient who's got minimal joint symptoms or had been treated for the Lyme and the joints got better, but they're having terrible brain fog and what we call CNS irritability, it's like it's this constant stimulus to the brain, almost like someone's overdosing on caffeine. There's anxiety, panic attacks, even rage attacks, and some very inappropriate antisocial behavior. Insomnia to a very strong degree. And the flip side is depression. The stimulation also can give you tremors, seizures, pseudo seizures, even a staggering, unsteady gait. So again, if you have a patient with known tick-borne disease or a chronic illness of any kind, where it's this enlarged, activated brain activity, you have to think of Bartonella. Now, Dr. Bransfield, who we know is great with his neuropsych background, theorizes that a lot of the people who are performing these antisocial acts and gun violence and so forth may have a brain infection, Bartonella, toxoplasmosis, so forth. In extreme cases, there can be hallucinations, even schizophrenia and dementia have been related to Bartonella. The other thing that's a clue are the skin manifestations. These so-called atypical stretch marks are now officially called Bartonella tracts, are a clue. I'll show you pictures. A similar infection of the skin is called bacillary angiomatosis. It looks like blood-filled pimples that can actually scab over. And if you biopsy these rashes, you'll find the Bartonella, and you also can often find some Borrelia mixed in. So the thinking is that by the time these rashes show up, which don't in everybody, but they do in some, you probably have an active Lyme infection as well as the Bartonella. There's not been a report of this in the literature, but the people who are great clinicians with Morgellons disease, or Morgellons, they say that if they don't treat the Bartonella along with Lyme, the Morgellons will not go away. And finally, there's a lot of literature on GI involvement. How many Lyme patients say, well, I know I have the joints in the brain and all that, but my stomach is bad, it hurts, it feels like I have an ulcer going on, my belly is sore all the time. Well, you have to think of Bartonella. Other things, in Lyme disease without Bartonella, I've found that most people wake up in the morning, the temperatures are below normal, like 96, 96.7, maybe 97. And as the day goes on, as they start to get through the day into the early afternoon, they start to have that big fatigue come over them. And at that point, they might have a low-grade fever of 99. With Bartonella, it seems to be the opposite. They wake up with a 99 fever, 99.6, so forth, and during the day, it tends to go down. Also with Bartonella, there can be some light night sweats, unlike Babesia, for example, the drenching sweats. But with Bartonella, there can be some sweatiness, mainly at night. It's light. General things, of course, a headache and fatigue. Neuropathy, again, because it involves the nervous system, the Bartonella, the neuropathy can be a prominent sign. So again, if you have a Lyme patient who's got neurologic symptoms way out of proportion to the rest of it, you have to think of Bartonella. Now, because Bartonella like to live in connective tissues, including the bone and also the skin around all this, you can find tender nodules. That's one of the clues that I've seen. For example, on the extensor surface of the arm, on the outer thighs, you run your hand down it, and if you find tender nodules along the outside, that's a sign of the Bartonella. Also, these same things can occur in the soles, and when people wake up in the morning, they start walking around, the soles are sore. So if you get up in the morning and you're going ouch, ouch, ouch for the first few steps, I mean, that's not a hard sign of Bartonella, but that's one of the things that's very much recognized. And in terms of joint involvement, you can have painful joints without the thickening of the synovial membranes like you would see with Lyme disease, because it's a tendinitis and a bone infection rather than an infection of the lining of the joint. See, the lining of the joint is called synovium, and when that gets infected, such as with Lyme disease or inflamed like with rheumatoid, that lining thickens up with scar tissue and inflammation. So because it's not a synovial infection with Bartonella, it's no swelling like that, but it's definitely painful. In the stomach, as I said, the GI can be involved. It can be the stomach itself with gastritis and heartburn, just like H. pylori. It can also be mid-abdominal pain, because the lymph glands which get infected by Bartonella can include the glands around the intestinal tract. That's called mesenteric adenitis. So it's usually the central abdominal pain that doesn't localize, and it just hurts. And it's not something that comes and goes. It's just part of the background. As I said before, the lymph nodes can be enlarged and they can be confused with lymphoma, as was with my patient. And I really wonder how many lymphoma patients really had Bartonella. And finally, another real big area of Bartonella involvement are the eyes. And the eyes are considered part of the central nervous system, so it's really not a surprise. But basically, any area of the eyeball can be inflamed and infected. It involves what's called a uveitis, even retinitis, inflammation of the retina, the back of the eye. And because Bartonella is in the bloodstream, has biofilm, can aggravate the lining of the blood vessels, the arteries and veins going to and from the eye can also become clotted, thrombosed. So every sort of eye involvement is possible with Bartonella. And again, we talk about this with Lyme disease, but I really wonder if you have a Lyme patient, terrible eye involvement, how much of that really is an unrecognized Bartonella co-infection. Another clue, unexpected response to Lyme treatments. Like I said before, very hard to pick out, unless one of these obvious symptoms are there, but very hard to pick out how many Lyme patients really have Bartonella as well. So think about a Lyme patient diagnosed with Lyme, given medication. One of the clues is that the Lyme treatments are not clearing the symptoms, because sometimes Lyme treatments don't work for Bartonella. Or on the other hand, the Lyme treatments may work to some degree, so the person gets improved to a certain degree, maybe 50 or 80% better, but there's that last percentage that just will not go away because Lyme meds will inhibit but not kill the Bartonella. Another very important clue that I used to use a lot in my practice, the Lyme germs really grow very slowly. And if you think about it, that's the reason why that Lyme has a 4-week cycle. The germs start to get active, then they calm down. It takes a whole month for them to regrow. So if you have Lyme disease, and let's say for some reason treatment was stopped too soon, either had a side effect and you had to stop it, or the clinician doesn't want you to take more drugs. If you have Lyme disease and stop, what happens is, interestingly, for the first few days, you might actually feel better. Herxheimer has calmed down, side effects of the drugs go away. But then as a week or two goes by, you're feeling all right, but then maybe by the 3rd, 4th week, you start to feel the Lyme come back because it takes so long for the Lyme germs to recover and grow. Bartonella, on the other hand, grows quickly. So if you have someone who's on Lyme treatment who's stopped abruptly before the treatment is complete, and they start to feel worse right away, then you have to start to think of Bartonella. And I remember patients would say, gee, I stopped the medication on a Friday, and by Thursday or Wednesday the next week, it was all back. I say, it can't be Lyme, because Lyme doesn't move that quickly. That's a clue of Bartonella. So that's one of the reasons why I have people keep a diary. Whether you're on antibiotics or you're stopping them, and especially when you're stopping them, how long does it take for the symptoms to come back? Is it a few days like with Bartonella, or a few weeks like with Lyme? Okay. Here's some articles on literature about basic patients, and this is pretty revealing. Patient 1 is a college student, cat scratch on her right hand, and she had, inflammation, infection, diagnosed with cat scratch disease. Probably wasn't treated right, because a year later she developed fatigue, headaches, memory loss, disorientation, poor coordination, insomnia, tremors, and infrequent pedimal seizures. So again, it's a neurological picture. She wasn't complaining of joint pain. She wasn't complaining of, the kind of brain fog you get with Lyme. You're talking about much more neurologic involvement. Patient 2 developed debilitating depression, insomnia, fatigue, loss of coordination, memory loss, disorientation, headache, and fluctuating severity that kept going on, at least for a year by the time the article was written. Third patient, someone who was, a jogger, a fitness person, got sick, started to stumble, had to stop jogging, got to the point they couldn't even walk without assistance, had to rest several times a day, and guess what? Diagnosed, or should I say misdiagnosed, as having multiple sclerosis. So very much a neurologic picture. Here's from a different article: fatigue, insomnia, memory loss and or disorientation, blurred vision, again the eye, loss of coordination, headaches, depression were the most commonly reported symptoms, seizures, severe paresis or paralysis, and debilitating migraines with predominant abnormalities in 3 patients, and 2 reported fatigue with accompanying neurological abnormalities that persisted for 3 to 5 years. And here's the article on the GI involvement. A pediatric gastroenterologist, Dr. Fried from New Jersey, wrote this article on 81 patients of his who had abdominal pain. They had abdominal pain, reflux, heartburn, and some even had blood in the stool. These were kids aged 8 to 21. And they took PCR testing of the biopsies that were done. And 33% they couldn't find anything by PCR, but 37% had one pathogen, 24% had two, and 8% had three different pathogens on the biopsy. Of the 81, 35 had Bartonella, and of these, none of them was seropositive. Now, this was back in 2004 when the testing for Bartonella really was not that great. But still, here we had people, 35 of them with Bartonella, not a single one had a positive test. And this Bartonella was bad enough to infect their GI tract, get them to a doctor where biopsies are being done. So that's a little clue about how hard it is for the clinicians and patients alike. And here's some pictures of the skin. These are the red bumps, bacillary angiomatosis. They look like pimples that get blood in them, and they can scab over. They can be painful. They can get red around them. Let's see if my pointer works. See the scabby thing, and then there's like a red halo around. Definitely not like an EM rash, but it looks almost like you had a bug bite or some irritation that you kept scratching and got infected. But this is actually from within coming to the surface. And these are pictures of the Bartonella tracts. Some people confuse these with stretch marks, but they're really not. First of all, stretch marks go in line with the plane of the skin. These are perpendicular to the plane of the skin. They don't follow the skin planes at all. Stretch marks are generally silvery and light. These are actually red. Sometimes they can be tender, usually they're not. But if you feel them, you can actually feel it like a little bit bumpy and rough on the surface because they're elevated and they're inflamed. And again, you biopsy these, you find inflammation, you find Bartonella. In some cases, or a lot of cases, you find Lyme Borrelia. Here's one under the arm. And one of the things you can see, here's one that's pretty straight, but this is kind of wiggly, serpiginous, they call it. It's crooked kind of a line. Stretch marks don't do that. Stretch marks are straight. Here's one on the back. I've shown this several times in my conferences, and it looks like someone's leaning back against like a lawn chair or something and left, indents in the skin. But look at how it goes here, and then it's broken up, and another one, all in irregular lines, because what happens is the Bartonella and the inflammation that it causes is dissolving away the connective tissue and the collagen and allowing the skin to separate and stretch and get kind of nasty. Here's one patient. He had both. He's got the dots, the spots, the bacillary angiomatosis, as well as the tracts. So they're all possible. Now, this is one of the things I want to talk about and actually emphasize. Bartonella is a major builder of biofilm. The biofilm is not only in the tissues like these slides show, but the biofilm is also in the bloodstream. Now in our blood cells have to circulate through some very fine capillaries to deliver the oxygen and nutrients and so forth. And the capillaries are very small, often smaller than some of the blood cells, which have to fold on themselves to get through. Well, I've seen slides of Bartonella biofilm circulating in the blood that was so large they contained many blood cells and many Bartonella and other organisms, all within this gel. And no way could this thick gelatin mass get through the fine capillaries. And I'm sure that's one of the real reasons why Bartonella causes so much pathology, because you're starving the tissues for blood supply, especially the brain, where the fine blood supply and transfer of nutrients is so very much important. All right, let's talk about Bartonella testing. As I showed you with the biopsies of the stomach, where 37 biopsies done on a single person in a positive serology, the traditional testing is really crappy. Crappy being a medical term again for the day. If you go to one of the big commercial labs for Bartonella testing, they do one Bartonella species, Bartonella hensley, a cat scratch. And as I showed you in the beginning, that we have at least 40 species we know about, maybe even more. The problem, though, is that if you were to say, well, if serologies are so bad, what if we do a PCR? A PCR is a direct test to look for evidence of the organism itself, rather than an indirect test like a serology looking for the immune system reaction to it. So the PCR is going to show up if there are a lot of Bartonella in the bloodstream, but often there isn't. And there's a reciprocal relationship between serology and the direct tests. And the reason is if you have a strong immune response, you might have a high serology and get a positive result, but a strong immune response brings the number of germs down low. So the direct test, which is PCR and the FISH test, may not pick it up. On the other hand, if there are a lot of Bartonella in the bloodstream, it's presume that the immune system's not working against it too well, and it's unlikely to have a positive serology, but then, because there are a lot more in the blood at that point, you can have a positive PCR of fish. So this reciprocal relationship, but we're talking about small numbers here. I mean, again, that article, 0 out of 37 were picked up. In my practice, using these older tests, we're lucky we had a 20% pickup rate. Likewise with the PCR, which is even worse. But newer tests have come out. One test is called the Immunoblot, came out by Igenix, and that's a kind of a serology that's much, much better, and I'll show you details on that. The fish test is a direct test where they actually take blood and put it on the slide, and they look at it under the microscope using special stains. You actually can see them, and I'll show you pictures. It's pretty cool. Galaxy Lab came out with an improvement in the PCR called the droplet digital polymerase chain reaction, the ddPCR, that increases the sensitivity of the PCR by a factor of 6. So that's really pretty good. And nowadays, you also can get a T-cell test called an ELISpot for Bartonella. And the beauty of that is that T-cell reactivity sometimes occurs even if the B cell or the serology is not positive. So you can have a seronegative, but T-cell positive. So it's one more way of looking at your body's reactivity to the presence of this infection. So like I said, the standard tests do badly. In America, we have IFA testing. The ELISA is present in Europe. An IFA test is really, really bad. I won't get into too much detail about it, but you have to look under a microscope and look for reactivity, color change, and it's so finicky. If you have a bulb in the microscope that's too old, it has a color change, it doesn't work. And labs that do IFA have a protocol that after so many hours, they have to put new bulbs in microscopes. I mean, it's really old-fashioned technology. The ELISA basically is that technology just made in an automated fashion, so it's really not that good either. Despite that, it's even worse because these are usually validated for one species. And if there are different types of serotypes within that species, just one serotype of that species. Most labs do the Bartonella-Henslier for cat scratch disease, as I said before. Some do Quintana, but it's just that, not any of the other 40. And if you look at the literature, sensitivities range from 28%, which is the most common number. Some say as high as 91%, and I don't think that's real. But if you look at it more carefully, those 91%, or the ones who had the swollen glands that were really obvious, had marked lymphadenopathy, or those with endocarditis where the heart valve is infected, spreading germs, showering the bloodstream with tons of germs. That's when these were picked up. But it's kind of circular reasoning because they said, oh, it's Bartonella because it's a positive test, therefore it's being picked up. No, it's going the other way around. How many of these people were undiagnosed because of bad testing? So again, we're talking about a sensitivity at best of maybe 28%. And here's a case series. There were 6 patients, all with severe neurological dysfunction. And eventually they were all PCR positive. So you know that they really, really had Bartonella. But patient 3, 6 times they were tested, all samples were negative. Patient 4 tested 3 times, all samples negative. Patient 5 was tested multiple times, only 1 out of them were positive. One of the other net ones was always negative, later on seroconverted. So again, we're talking about really bad sensitivity by a standard serology. The flip side is actually even worse. The test is so non-specific. Look at this, a false positive is reported anywhere from 27 to 68% of the time. So that means in some cases, more than half, almost 2/3 of these cases where you have a positive Bartonella test, it wasn't Bartonella at all. For example, this one article that's down here, I'm pointing to the screen, here's the arrow. This one series, 36% of positive Bartonella serologies were not from Bartonella, but from a chlamydia infection. 24% were from Epstein-Barr, cytomegalovirus, Coxiella, even strep gave a false positive on a Bartonella test. So from a different series, tularemia and even toxoplasmosis can give you a false positive. So these Bartonella tests are really very bad, these serologies. And why? Again, familiar story. So many species, 13 of which we know exist here in America for our patients. And the standard serology is validated for 1 species and only 1 serotype of that species. And one of the articles mentions that there's a regional distribution of different Bartonella. In other words, Bartonella in the South may be different from those in the Midwest, in the North, in the mountain regions, in the valleys. They're all different because they have different hosts and different vectors. So if you have a lab that's looking for 1 type of 1 species, how accurate can that be? Also, sometimes the serology just won't pick it up, even if you have the right test for the right germ, because the Bartonella can evade the immune system by hiding inside cells, by hiding inside biofilms, and so forth. So if you have a serology test that's not very sensitive, and then the germ is hiding, this so-called stealth infection, of course, it's going to be a very low pickup. Now, with serologies, there's also a trade-off between sensitivity and specificity. So as I showed you before, it's not a very sensitive test. So what if you turn up the volume? So you say, well, we'll start to make it more sensitive to maybe pick up more of them. Well, then what happens is it gets so sensitive that you end up picking up things that aren't Bartonella, like I showed you the false positives. So they have what's called a cutoff between a positive and a negative. And the way they determine that cutoff is a statistical analysis. And if they lower the cutoff, making the test more sensitive, picking up more germs, it gets less specific because it also picks up things it shouldn't. So they say, I don't do that. Let's make it so it's very specific. And if we get a positive, we know it's Bartonella, but then you're going to lose sensitivity and miss a bunch of them. So it's this bad trade-off with serologies. So that's why the immunoblot has replaced them all, because it's completely different technology. Standard serology, be it an ELISA or an IFA, even a Western blot, they take the Bartonella germs and they grind them up and they powder them and they make the test out of these ground up germs. So germs have a lot of proteins in them that are not specific to that germ. So if you make a test from that, you're going to pick up other germs. If you try and purify it to the point where you have very narrow range of germs from the Bartonella, well, different Bartonella have different sets of germs. You're going to miss some of them too. So what the immunoblot does, which is totally different, they don't make the test from Bartonella germs. They take genetic sequences of Bartonella that are known to produce Bartonella specific proteins, and they use a virus vector to transfer these from Bartonella into an E. coli, and the E. coli germ produces these in large quantities in a very highly purified fashion, and they use that to make the test. So it's kind of like reverse engineering. They go not from the germ to the lab, but from the genetics of the germ to produce the exact protein they want, and then make the test out of that. So the beauty here is that this trade-off between sensitivity and specificity is gone, because you can make it as sensitive as you want without sacrificing it being specific, because you're using exactly the germ proteins that you want and nothing that can cross-react with another germ. You can also choose these recombinant things from a number of different Bartonella species, so you can get a whole bunch of different ones, not just the one that the commercial labs use. You can also do it so that you pick up Bartonella by genus, not species. So it's like Bartonella cantana, Bartonella hensleyi, and so forth. Well, if you just say, let's do just Bartonella period, and any Bartonella will be picked up, that's what's called genus specific. And that allows you much broader coverage of all the different Bartonella. And also an immunoblot can be engineered to show both IgM and IgG, and that makes it more sensitive. So again, recombinant antigens are synthetic ones created in the lab, specific for Bartonella. So it's much less likely to pick up non-Bartonella proteins and is much more sensitive at the same time. No longer cross-reactivity, no longer this trade-off of sensitivity, specificity. And this immunoblot that we now have commercially available, it'll pick up pretty much all the different species that are of interest to the clinicians and the patients. And these several that are here, hensleyi, cantana, elizabethae, and so forth, if they're present, they'll actually be named. So if Bartonella in general found, so we found Bartonella, and in addition, we found these specific ones. So it's really neat to help figure it out. And this is also where you can find that there are actual co-infections. So how good is it? Well, there's 15 samples, for example, one of the series of validation that were definitely positive for Bartonella, proven by PCR with sequencing. All of them, 100% were positive by the Bartonella test, by the immunoblot Bartonella test. So that's a lot different from what you saw in the other one. How about the specificity? Are you going to cross-react to Toxo or some other thing? Well, they did here a case of 34 where there were some positives and some negatives, and the specificity of this was, again, 100%. So there's no cross-reactivity with the other germs. Here, for example, looking for co-infections, 5 patients with Lyme disease, none of them showed up on the Bartonella test. People with Lyme and relapsing fever Borrelia, none of them picked up on the Bartonella test. Lyme and Babesia, Lyme and Anaplasma, Babesia alone, even some negative controls from other diseases and healthy people, none of them showed a false positive on the Bartonella test by immunoblot. And here's a validation study. In addition to what I just told you, they had patients who were positive for Bartonella by any variety of other tests IFA, fish, Western blot, so forth. And if they were positive on that, they were positive on the immunoblot. So putting it together, again, the specificity is 100%. So in other words, if you have a positive on the immunoblot, you know you have Bartonella. It's not like you have some kind of a virus that's giving a false reading. And sensitivity, IgM and IgG, 75 and 89%. But if you say it's one or the other or both, then it came out to be 100% were picked up on this. So that's really pretty good. Now, what about a PCR? The standard PCR, as I said, the pickup rate is really very poor. Here's an article where they found only 6 out of 112 samples were positive, and that's a 5% sensitivity. So that's, like I said, what we've seen in the world, in the real world. Here's what Galaxy did. They did drop the digital PCR that gave it 6-fold more sensitivity, up to 30% sensitivity. They actually raised it up to as high as 42% by having 3 separate blood tests run 2 weeks apart, where they pooled it together and cultured it for a month, and then they did the test. Of course, that means you won't get your result for 2 or 3 months after the fact. Then they did pick up the sensitivity to as high as 42%. So, labs are jumping through hoops to try and get the diagnosis better. The FISH test, that's a favorite of mine. And why? Well, first of all, FISH is a fluorescent in situ hybridization. I like that. What this does is it detects the RNA of the pathogen, not the DNA, but the RNA, and you can get a positive or negative on this. It's not a titer. It's either yes, you have it, or no, you don't. And what they do is they take a blood smear, blast slide, they put a drop of your blood on it, and they flatten it out. And after it dries, they stain it with stains that hybridize or attach to the RNA in a very specific fashion. And then it's tagged to a fluorescent dye. So they look under the microscope using a black light. That's like back to the 70s. And so it has a very high pickup rate. If it's in the bloodstream, they have a very good chance of picking it up. And the interesting thing about RNA is that RNA doesn't stick around. It has a very short half-life in the blood, matter of hours after the organism disappears from treatment. RNA disappears too, because it's produced by a living germ and disappears when the germ dies. So positive on a FISH means the time the blood was taken, within hours, that person had an active infection. But the thing that makes me excited about this, and why it's one of my favorite tests, is that it can detect Bartonella even if it's in a biofilm. This is where FISH testing got famous, because of industrial testing. There are many things like water treatment plants and other places that get contaminated with biofilm building organisms. And the only way they could find if these organisms were present to the biofilm is by developing a FISH test for it. So this technology has now been applied to tick-borne diseases. We have a Babesia fish, now there's a Bartonella fish, because it can find them even in the biofilm. And here again, the RNA is hybridized at the genus level. So the different species can be picked up. They've proven it for the ones listed here, Helmsley, Quintana, Vinsonia, so forth, that they've proven. Others are possible as well, because again, it's a genus level test. Here's some nice pictures. Here's Hensley A, here's my arrow, there it goes. Hensley A, Quintana, Elizabethae, negative control, nothing. So, if you have a microscope and you're scanning it and you see this, you see it, there it is. And because it's very specific, you're not going to find these little glowing things in someone who doesn't have Bartonella. And here are actual patient samples. This is a Hensley A, the first and the second one, Quintana, the third one, and here's a negative control. And the positive, by the way, were confirmed by DNA PCR with DNA sequencing. So there's no question there about it. And here's a performance of the FISH. Specificity study, 100% specific. In other words, none of these other things gave a false positive. They include two different Babesias, Borrelia, Trypanosoma, which is a parasite. This is malaria, Anaplasma, Ehrlichia, different rickettsias. No cross-reactivity to those. But on the other hand, blood samples from patients that were PCR positive, confirmed by DNA sequencing for Vinsonia, Hensley, Quintana, and the other ones, even one that wasn't able to be speciated was definitely a Bartonella, though it picked up every single one of them. So here again, we have a test that's 100% sensitive, 100% specific when the germs are present in the bloodstream. And finally, the T-cell testing, totally different method. And one of the things that, as I mentioned earlier, to really pick this up, you want to use testing using different methods. So you're not going to do one test on a patient, you're going to do a combination of tests. But silly to do 3 different IFAs or ELISAs, you want to do an immunoblot for the serology, you want to do a PCR or a FISH test for direct, and you want to do a T-cell response for the T-cell end of it. Because T-cells respond independently of B cells. This T cell test is, again, genus specific, so it can pick up a wide variety of species. T cell testing is interesting because there are certain time sequences of the illness where the T cell test works. One is very early in disease, within a week of exposure, lasting for about the first 6 weeks, is when it's really very sensitive. After that, the sensitivity drops off until it gets a very late disease when someone's really chronically sick, then it becomes sensitive again. So if you use this in the right timing of the illness, you find it to be very useful. And because T cells are independent of B cells, you can be seronegative, which is a B cell thing, but be T cell positive. And that's again why T cell testing included with B cell and direct testing is a really good way to go. And finally, a T cell is part of our immune system. That's a T cell response test. A B cell is the other part of our immune system. That's a serologic test. So if you do a serologic test, especially a sensitive one like an immunoblot, and you do a T cell test like this, and then you do some direct test, let's say a fish. What if the fish shows that the Bartonella is present, but the T cell test is negative and the B cell, the serology, is negative? Well, that not only means that, well, you believe the positive and ignore the negatives, but also means that something is wrong with the immune system. In other words, the T cells are not recognizing this thing, the B cells are not recognizing this, but it's there by the fish. So this is an independent way of not only making a diagnosis, but also helping you to figure out whether or not your immune system is up and functioning. It's a pretty cool trick. And again, that's why you want to do a panel approach. You want to do B cell testing, you want to do T cell testing, you want to do direct testing. Now, what if you do 2 or 3 or 4 of these tests and 1 or 2 of them are positive and all the rest are negative? Well, the tests that I've mentioned, the immunoblot, the fish, the PCR, they're all very, very specific for this germ. So you believe the positive and you can ignore the negative, okay? So this is one of the cases where it's not like, gee, 50-50. No, the positive you believe, the negative you don't. All right, treatment. Now, treating Bartonella is so involved and so in-depth because everybody is different and because the germ is nasty. So we'll go through the problems. First of all, as I said, it could be in the blood, could be in the tissues, could be in the cells, out of the cells. The problem is not every antibiotic works in all these different spaces, so you end up having to use combinations. As I mentioned, biofilms protect the Bartonella from the meds and from immune attack. Bartonella also can go dormant, just like Lyme disease. It's a form of hibernation called a stationary phase. They're still alive, but they're not growing, they're not dividing, and antibiotics kill germs while they're growing. So if you have a germ that's not growing, it's just sitting there dormant, what are you going to do? Nothing. Well, there's some tricks we'll show you. Other thing is that besides all the different species I mentioned and different strains of the species, each species may have its own separate antibiotic susceptibility panel. So what works for one Bartonella may not work for the other. And since you can be co-infected with more than one, now it gets even more complicated. All right, let's do some studies. Well, the problem is most of the studies, if you look in literature, they take the Bartonella, put it in a petri dish, and they test against drugs. Say, wow, this one works great, and this one's fine. But the trouble is Bartonella don't live in petri dishes. They live inside our tissues, inside our cells. And what happens in the petri dish is very much different than what happens if a bug is living inside of our monocyte or endothelial cell or a muscle cell. So that is why the drug studies are not very good and why treatment has never been that great until more recently. Also, how long do you treat Bartonella? There's no way to tell when the thing is gone and the symptoms are so vague, so who knows? But we have some hope. First of all, they found that rifampin, erythromycin, azithromycin, doxy, and cipro, while they were good for growing Bartonella, they did very poorly against the stationary phase dormant ones. Daptomycin and methylene blue and gentamicin, they were able to eradicate the stationary phase. So that sounds really pretty good. On the list here is also streptomycin, which you don't really use because it's toxic and has to be given by a very painful injection into the muscle and the tuchus. And clotrimazole worked, but clotrimazole is an oral anti-yeast troche that you dissolve in your mouth. It doesn't absorb into the bloodstream. There's no way of getting it IV or IM. So those are really not on the list. That's why I didn't emphasize them with the bold type. But anyway, so daptomycin, methylene blue, and gentamicin, that seems to work. And here what they did was they tried 14 different antibiotics and 25 combinations to see what will work against a stationary dormant one. They found cipro and gentamicin, even nitrofurantoin were the most active, cipro and gentamicin. Methylene blue and rifampin were very good against the biofilm form, and they found combinations of azithro and cipro, azithro and methylene blue, rifampin and cipro, rifampin and methylene blue, they could kill the stationary phase and the biofilm phase. So wow, that sounds actually pretty good, but not so fast, okay? Again, that's studies in a test tube. Here they found that erythromycin resistance is coded for in the genetics of the Bartonella germ. They found that this mutation is the most frequent mutation found in it. And this is, they say, this is why treatment failures and relapses are so common when using azithromycin. And for Cipro, which is a fluoroquinolone, all Bartonella isolates became resistant to the fluoroquinolone after differing numbers of cultures through their test tubes. So what looked great may not be so great. So now let's go through the list in detail. There's cell wall drugs like cephalosporins, augmentin, which is augmented penicillin. In the test tube, they will kill living Bartonella, the active growing Bartonella totally, but these work in the intravascular space, not in the cells, and they may not be so great in the tissues. Also, the Bartonella have enzymes called beta-lactamases that can dissolve these things. So you have to have a cephalosporin or a penicillin like this augmentin that's resistant to lactamase. So cell wall drugs might work in part of the infection, the part that's in the bloodstream, but not for the tissue ones. The fluoroquinolones like the Cipro, the Levaquin, and so forth, that has very good sensitivity. However, as I mentioned before, they can become resistant to it over time. And as many of you know, fluoroquinolones for some people can have toxic effects in the central nervous system with irritability and insomnia and anxiety, and also can cause semi-permanent tendon side effects where the tendons really get inflamed. So that's a limiting factor. And some people will try this carefully, so others will say, I'm not going to go there. Macrolides like azithromycin, they seem to work great, except rapid development of resistance. And here's the key as a single agent. And rifampin seems to work for a whole bunch of different phases of Bartonella. Turn off my phone. But number one, it's weak. It's bacteriostatic. In other words, it inhibits the germ. It doesn't kill it. It's not bactericidal. It also induces the cytochromes in our liver, which means that other drugs that we have in our system, even herbs, they interact. They may be eliminated more quickly from the body. They may be made more potent. So that's a problem. And because our hormones are also eliminated through our body by cytochrome system, a lot of people have a temporary adrenal insufficiency while on rifampin, even a thyroid deficiency. So that has to be attended to also. How about Bartonella for tetracyclines? There's a famous quote from Dr. Breitschwert, who, as you all know, is the genius on Bartonella. He says, you cannot float a cat, a dog, a horse, or a human in enough doxycycline to eliminate Bartonella hensleyi. I visualize a bathtub full of doxycycline, dipping the patient in, and it's still not working. Now, how about gentamicin? In every test in the test tube, it seemed to be the best drug. However, and again, it sounds good. In this one series, patients receiving an aminoglycoside, which is gentamicin, were more likely to fully recover. And those treated with this for at least 14 days were more likely to survive than those with the shorter treatment. These are patients with a heart infection, which is like the worst case. However, gentamicin does not penetrate the central nervous system. For some other infections where this has to be used, for brain infections, actually have to give it through a spinal tap, which I don't think we're going to do for a Bartonella patient. And this can be toxic to the kidneys and to the ears too, especially if combined with a macrolide like azithromycin, affects the ears and the kidneys. Now, methylene blue is an agent that has been recently found to be effective, and there's articles on that. And rather than try and go through the details, I've got this quote from one of our practitioners. And the person said, I use methylene blue routinely with not by itself, but with Zithromax, Bactrim, rifampin, and so forth, not all at once. They have to check for G6PD, which is a genetic inborn metabolic, I won't call it a defect, but it's a metabolic trait that if you have this gene, it makes methylene blue toxic. So you have to test G6PD deficiency before you do it. They also mix methylene blue with Bartonella herbs, such as A-Bart, Bar1 and Bar2, BLT, oregano, berberine, biocidin, and so forth. This person calls it a major game changer. Start low and titrate up to 100 to 150 milligrams twice a day, but it causes severe oxidative stress, so you have to be sure that the patients are on antioxidants, and various compounding pharmacies are the source for this. So, methylene blue might be a game changer in some people's hands, but that's the key. You have to have someone who's used it before, who knows about it, knows about the side effects, knows what to look for. And finally, the treatment duration. This is from an article that came from Europe, actually. And this person said, when the treatment duration is less than 15 days, relapses are common. Therefore, treatment must be given for 3 to 4 months. Now, this is interesting to me because this was written based on Bartonella infections of the heart and other severe infections of Bartonella. And it's from Europe and it's fairly recent. And decades ago, when I started working with Bartonella treatments and working out the methods, I also found you need a good 4 months at a minimum to really get rid of Bartonella. And here they found it themselves. So they listened to me. No. All right. Before we close, I want to talk about supplements. There are things called transfer factors. Some of the labs, some of the supplement companies make this. It's been found that these are really effective in Bartonella. Because remember, Bartonella, as I mentioned, can actually turn on autoimmunity. Well, transfer factors can actually stop that from happening. They also produced in our immune system to stimulate the production of new help or making new helper T cells, new killer cells, macrophages, and cytotoxic T cells. So again, the reference is here, but transfer factors have a huge and important role to take while treating Bartonella. Now, here's an article where one of my colleagues put together a great study to see which herbs are proven to be toxic to both the growing and the hibernating stationary phase Bartonella. Cryptolepis, black walnut, Japanese knotweed, berberine, and acai also were found to be very helpful to kill them. And it was in concentrations that you can achieve by taking these supplements orally. Another one very important was not part of that, but a different study, arginine. If you can take arginine and not have herpes reactivate, which is one of the side effects, well, they found that arginine counteracts the negative effects of what Bartonella does to our body through nitric oxide. And that's what starts the inflammatory cascade in Bartonella. So if that gets inhibited, that goes a long way toward making the infection less damaging. Also, they found, and this is really, really interesting, that by taking the arginine, it stops the genetic change in Bartonella that makes them drug resistant. So if you, again, can take arginine and not reactivate a latent herpes or have enough lysine on board to prevent this from happening, this is something you want to get on in the beginning of treatment so you don't get drug resistance. Really neat clue. Other things, mitochondrial rescue. Always have to do that first. Chronic illness of any kind, chronic inflammation of any kind, inhibits the function of our mitochondria. Mitochondria produce the energy of our cells, and our immune system is vitally dependent on super duper doses of energy made by our cells. So if you don't have good mitochondrial function, you're not going to go anywhere. It starts by detoxifying. What does that mean? The toxins that we build up, like insecticides, glyphosate, heavy metals, what they physically do is they bind to the membranes inside the mitochondria and prevent the steps of metabolism that produce energy. So you have to get the toxins out. You have to support the membranes by putting new factors in, NT factor, phosphatidylcholine, omega-3 and 6, to build back the good stuff in the membranes and crowd out the toxins. Then you support the metabolism with the B vitamins, CoQ10, NADH, lipoic acid, methylation support. A number of you have heard about methylation support because they might have an MTHFR defect. Trouble is, that's not important. I say that because everyone with chronic infections in the tick-borne field have methylation defects, even if the genetics are fine. But the process gets inhibited regardless of your genetics. Even if your genetics are fine, you've got to do methylation support. Now, just to give you a very simple regimen, what I have done for my own Bartonella when I had it, Research Nutritionals has 3 different products, and I like this combination. I'm not advertising for them, it's just that I take them myself. Their Physicians Daily is a multivitamin. ATP 360 is meant to support the mitochondria, and Energy Multiplex supports the rest of the metabolic chain. And these 3 together will make a difference that you can feel. And they're not terribly expensive, they're easy to take, I think anyway. They don't bother my stomach. Next thing is you have to address inflammation. Because when we have inflammation, that activates the germ and allows it to start to do some damage. What are anti-inflammatories? Modified citrus pectin is a very important one. Curcumin, all those others, resveratrol, these modify the inflammatory response and moderate it. And after these things are started, then you can start to work on antimicrobial herbs and the botanicals. There's specific formulas from Research Nutritionals, Beyond Balance, Burr and White. You begin at very, very low doses and watch out for herxoma reactions. And sometimes they're very strong. I know with Burr and White and Beyond Balance, we have to start with a drop or a half a drop or a quarter of a drop every few days. How you take a quarter drop, you put the drop in some water and pour out 3/4 of it. Just because the herxomas can be pretty wicked. I also found that as you go through this and get the doses higher and higher and higher, suddenly they become intolerable. And that's because you've got to the point where you don't need it anymore. That's a very strange response, but that's what happens. All right, I'm just about out of time, so we'll conclude. Diagnosing Bartonella can be really, really difficult. You have to rely on clinical suspicion and not one, but a panel approach to testing, where you get different tests using different methods, all done at the same time, and you believe the positives and don't believe the negatives. The treatment is not straightforward because you might be treating more than one species at the same time. You have to, again, use combinations of antibiotics. Other antimicrobials like the herbal ones, methylene blue, you may want to add on. Again, your doctor has to be familiar with this and comfortable with this and aware of the side effects and how to manage it. You have to be alert for the development of drug resistance, because that definitely happens, and you have to sometimes just rotate through different medicines. And be alert for recurrences and relapses. And remember the time course. Like I said, you stop the medicine, you feel worse right away, the Bartonella is back. The other thing is you can treat this concurrently when you treat other tick-borne diseases, because there is overlap of medications and the herbals and the supplements. And as I said, there's a good role that's now been documented in scientific literature that the complementary therapies actually can work. So there we have it. I thank you. Now, I think what we're going to do is what? Going to start chatting?
Yeah, we do have a couple questions, Dr. Burrascano.
Okay.
First one is from Tara. If someone does not hurt, would that mean these therapies aren't working?
Well, I don't know. If you're on a regimen that's very, very gentle and mild because you're starting low and you're not herxing, well, that's actually a good thing, because herxoma reactions, I mean, we look for them, but they're not necessarily good for you. I mean, they are causing more inflammation, which in the long run, or at least in the short run, can make things worse, both germ-wise and damage-wise. So you don't really want a strong herxoma reaction. But if you start gently and increase, and you're not feeling better and you're not herxing, then yeah, I think it's not working.
Okay, we have a question from Medi. Curious about the last comment, when a high dose of Beyond Balance or other drops cause a bad effect when you don't need them anymore?
Oh, that's a really interesting thing I found. Let's say you start with like 2 drops twice a day, and over the next few weeks, you go to 4 drops and 8 drops, and you find up to 20, 30, 40 drops, a whole dropper full twice a day. And you're feeling pretty well. And I'm talking about when you're through the treatment and you're almost back to normal or are back to normal. And you start to feel like every time I take those things, I'm feeling rotten. I'm feeling tight breath. I'm feeling tired and achy. My head is hurting. I'm going to stop them and see what happens, and stop them and say, wow, that was what was going on. Here you were maybe on weeks or months of this treatment. It was gradually making you better and better and better. And suddenly it's making you worse. I think that's when you get to the point where you don't need them anymore, or at least you stop. And if you still need them because you're not completely over the illness, number one, you have to think that there's a co-infection that's not being treated. And number two, then you might think, all right, if I'm going on Beyond Balance, this is what's happening, let me go over to Burr and White. I've seen the same thing with Burr and White, that it gets up to the point where, oh, I can't take that anymore. And then you can go to Beyond Balance and you're fine with that and you can go up on that. But these herbs, I don't know if many people realize it, but these compounded herbs from those 2 companies, they don't talk about this because it's kind of, thought by many to be fringe medicine. But they work not so much chemically through your body like drugs do. They work energetically through the energy balance of the system. And that's what's been thought about. I know there's no way to really prove it, but that's the theory behind it. And apparently you get to the point where your body has had enough of it and you're ready to change to something else. And it's had enough either because it doesn't need it anymore, the germs are not there to soak it up, or because it's done as much as it can do and you have to move to another one. But that's a very interesting thing I found with both of those formulas, both of those brands. And it's kind of a common thing, I think. So it's a clue. I like these clues.
Okay, next question is from Ben. What do you think of Dr. Moziani's protocol consisting of clarithromycin and rifabutin?
Well, rifabutin is a different form of rifampin. And as I said in the previous lecture, using a macrolide plus a rifampin derivative can work against biofilm and stationary phase together in some people. But rifabutin is, like I said, bacteriostatic, not bactericidal, and macrolides can get drug resistance. So that's one of the subtleties and problems with treating Bartonella in general. If you're going to use a medicine like a fluoroquinolone or a macrolide that can rapidly develop drug resistance, that happens not immediately, but over time. And the way you can help to get around that is by using combination therapy and by using a high dose. So here you're stuck with the dilemma of having to use combination therapy at a relatively high dose right away in the beginning. You can't sort of ease into treatment, because if you ease into treatment to minimize Herxheimer and get the person to tolerate it, you run the risk of allowing the germ to start to resist it, because you kill the strong ones. You kill the weak ones and you leave the strong ones behind. So that's a dilemma where you want to put them in right away and then hopefully not have Herxheimer and have to back off. On the other hand, that is also a way well, let me put it the other way. That's also one of the reasons why we do what's called pulse therapy. We use a strong dose of combination therapy right away, induce a Herxheimer, and then stop. And then let the body calm down, then you do it again, and then you stop. This is a way of keeping them out of dormancy, keeping them out of the hibernation, and using a strong dose that's not going to develop drug resistance. Very hard on the person, very tough to kind of figure out to schedule this. But it's a tough germ, and if you have an experienced clinician, that's what you have to do.
Okay, next question is from Claire. Thank you, Dr. B. Does having an active Bartonella predispose one to getting long haul COVID?
Oh, I think so, absolutely, because I keep close contact with my clinician friends who take care of tick-borne diseases and now long COVID. And I know in one office in particular, 100% of the long haul patients that she's seen test positive for active Bartonella. So if you have Bartonella, which can go dormant and you can live with it, if you're healthy otherwise, and get an infection like COVID, which is known to inhibit T cell responses against intracellular organisms like Bartonella, it's part of the problem. And, the Lyme and COVID practitioners who know about this are way ahead of those who don't. I mean, you hear in the lay press all the time about the problem of long COVID and their clinics and all that. They're trying all sorts of crazy things, but they're not trying things that make sense because they don't know about tick-borne diseases. So yeah, Bartonella and long COVID is a real big combination, very common, I think.
Okay, next question is from Amy. On a different topic, have you had any successful treatment for Lyme patients with COVID vaccine side effects?
What I briefly mentioned, I'll go into more detail now. The article that I'm referring to showed that a COVID infection or a COVID vaccine, either one, will change the balance of the immune system, where certain components of the immune system are made stronger and certain components of the immune system are made weaker. What's made stronger, paradoxically, is the response against molds and yeasts and candida. At the same time, antiviral immunity, especially the DNA viruses like Epstein-Barr and cytomegalovirus and so forth, is weakened, as is immunity against intracellular organisms, the stealth organisms like Bartonella. So let's say you have a long COVID patient who's also been tick-borne disease patient. This person probably has overgrowth of yeast and probably has Bartonella and Borrelia and all the others. And then they get either the vaccine or COVID or both. And then they start to get sicker. Part of that is the outcoming of these latent germs, but part of it also is they may be herxing from killing candida that wasn't able to be killed before. So it's a very hard thing for the clinicians to try and wind the way through and thread the needle. Are you feeling worse from a Herxheimer from killing candida or from the germs coming out, or a combination of the two? And the management of that is really hard. It's patient by patient. You have to see what's going on. There's no way to tell if the reaction is a Herx from candida versus a worsening from Epstein-Barr coming out. There's no test for that. It's all seat of the pants.
Okay, next question is from Jenny. Dr. B, have you used LymePlex Pro? It made me Herx bad. Yes, I am still testing positive.
I've not used that. No, I don't know what that is. Sorry.
Okay. Okay. Nanette, this is from Nanette. Can Bartonella cause darkened skin on toes and elbows?
I've not heard of that. I really haven't heard of that. I think it's something that I would love to have you get biopsied by and analyzed by Dr. Mosiani, because he's the one who's been doing these biopsies and doing some beautiful pictures of what's actually happening in the skin of the Bartonella patients. And he's the one who's biopsied the tracts as well as the other lesions. And that would be a great thing to look for, because to some extent, it sounds like an ACA lesion, like you see in chronic Lyme with the European Borrelia, where you see on the toes and elbows the darkening and thinning of the skin and more prominent blood vessels. But Bartonella is part of that too. So I would think that if it's anything tick-borne related, it would be from one of the European Borrelia. And that's why you want to do an immunoblot biogenics to see what's going on, and also get your skin biopsied. And it's a very simple skin punch biopsy. Any dermatologist can do it. You just have to send it to Dr. Mosiani's lab.
Okay, and then this is our last question. We're out of time. This is from Brad. I read your article in this month's Townsend letter about removing yeast from the digestive system. You recommend Clorox bleach as an antiseptic used orally. As a substitute, could MMS/chlorine dioxide be used? Also, how often and for how long should this oral treatment be used?
MMS chlorine dioxide has been, fluorine dioxide has been actually on the market for this for a long time. And I've not used it because this is so simple, so cheap. And actually, I'll give you a quick story. I know we're out of time, but I had a patient years and years ago who had such thick growth of yeast on her tongue, it looked like fur growing on it. I mean, it was really not nice. And we gave her Mycelex troches, and we gave her fluconazole, and she had a tongue scraper, and all these different things you can think of. Nothing. One day she came to my office and her tongue was perfectly fine. I said, "What was that? What'd you do?" She laughed. She said, "I used Clorox." It's like, whoa, she did it. Anyway, so I looked it up, and sure, there's a way of using Clorox where you dilute it very much with water. It's called a modified Dakin solution. A non-modified Dakin has things like potassium carbonate in it to make it a little bit less acidic. But anyway, you can get away without using that by diluting it enough. And it works. Basically, you put a small amount of this very dilute mixture. It's one teaspoon, 5 cc's, and 4 ounces of water. Put a little bit in your mouth, you swish and spit, never swallow it. Put a little bit more in your mouth, and you brush your tongue, your teeth, your gums, everything inside the cheeks with a toothbrush while you're holding it in your mouth. You spit that out, then you use plain water. Once again, you scrub it out. And now all the germs are gone, not just the bad germs, but the good germs too. And the problem is the yeast are going to come right back because they're opportunists. So the thing you do immediately is oral acidophilus, a mouthful of yogurt, something to put the good germs back in so the yeast can't come back. And Research Nutritionals just came out. I had talked to Dennis from Research about this years ago. I said, "You got to come up with something for my Clorox patients." And so he finally did. It's a little tablet you dissolve in your mouth. It's an oral form of acidophilus meant to coat the tongue and put the germs back. So I said, "I got to write this up." So I wrote it in Townsend. I'm glad it's out there. I haven't even seen it myself yet, but I'm glad it's out there. So yeah, it's a good thing. And in terms of using another one, I suppose you can. I don't have experience with it. And why spend the money? I mean, a whole bottle of Clorox costs a few dollars, will last you probably your lifetime. The question is, how often do you do it? Certainly no more than once a week. If it's coming back within a week, then there's something else going on. You have to build up your immune system or watch your carbohydrates or better oral hygiene or something. But it's not meant to be a daily therapy. And to be honest, I don't know if it does anything terrible for your teeth in terms of dissolving away the enamel or something. I just don't know. So that's why you keep it nice and dilute. You spit it out, you rinse it out, and don't do it that often. A little bit of, what should I say, backyard science here.
And then we had one question. Are you taking new patients, Dr. Burrascano?
No, right now I'm not. The way I'm working is through other physicians who need my help. They consult with me and I manage the patients through them. But I personally am not seeing patients now, no.
Okay. All right. Great. Thank you so much for joining us. We appreciate it.
Well, thank you for giving me the time. I enjoy speaking with you guys. And I think the more you as patients, the better off you're going to be. And then you spread the word. So again, I thank you. I have to give you back to screen, right? How do I stop that? Do this?
Yeah, there you
There we
go.
go.
Oh,
There you go. There you go. Thank you so much, Dr. Burrascano.
Again, you're welcome.
All right. So let's turn this over now to Dr. Kinderliner. And he is going to be speaking with us today about a toolkit for promoting your own healing. Dr. A. Kinderliner, MD, is a nationally recognized physician with expertise in the fields of nutrition, allergy, environmental medicine, Lyme disease, and the healing of mind, body, spirit as a unified whole. Dr. Kinderlehrer is a co-founder of the New England Center for Holistic Medicine in Newbury, Massachusetts, and has taught extensively, including practitioner training courses at the Omega Institute, the National Institute of Behavioral Medicine, the American Academy of Environmental Medicine, and the International Lyme and Associated Disease Society. He created and organized the Lyme Fundamental Courses, which is presented annually at the International Lyme and Associated Disease Society Conference. He is the author of many articles in the Medical Journal and in the Lyme Times. His integrated medical practice is here in our backyard, Denver, Colorado, and he focuses on the diagnosis and treatment of tick-borne illnesses. Dr. Kinderlehrer, is the author of Recovering from Lyme Disease, the Integrative Medicine Guide to the Diagnosis and Treatment of Tick-Borne Illnesses. This was released in March 2021. Thank you so much for being with us, Dr. K. Handing this over to you.
Thanks so much, Jody. And thanks to Joanne, Jody, Monica for making this happen. I know it's a lot of work, but I think it's a great service. And thank you, Joe. You always do a fantastic job. I just want to say I'm personally indebted to Joe. He was he continues to be one of my mentors. Someone I call on, and he's just a great colleague. And always a wealth of information and a wonderful attitude. So, okay, I am going to pull up my PowerPoint. Oh, geez, let's see. Hold on. Allow Zoom to share your screen. You know, this is, let's see, I think we got it. We got it.
All right, good. Okay, good.
Oh, it did something weird on me. Okay, so a toolkit for promoting your own healing. You're probably wondering, what's this about? I will explain shortly after this slide. Here we're going through airport security. This is my wife, by the way. Lyme disease, huh?
Hey, Dr. K, I'm so sorry to interrupt you. I'm not seeing your, I'm not seeing it.
Eesh. Okay.
So sorry.
Neither.
Okay. Wow.
I'm gonna make you, I'm making you a co-host just in case that is what we need to do. So let me try that.
Okay.
And now give it a shot.
Okay, let's see. This is, let's see if this does it. Is anything coming up now?
No.
Okay, right now, Jody, what I'm looking at is a few dozen people, okay? Just their names. Usually I see at this point just myself, because when I try to share the screen, what's coming up is very different from what I usually see. I'm not seeing, usually it goes to just a home thing, and then I can choose my PowerPoint.
If you look at the bottom of your screen, can you see a green icon that says share screen?
Yes,
Hit that.
That's what I'm doing.
And it's still not letting you, huh?
Well, when I hit, it's different. Like I said, this looks different. It says Microsoft PowerPoint, great. And then it's giving me the option. It says open system preferences. I'm not sure.
That is odd.
Very odd.
Do you want to just quickly send it to me and I can share it while you talk through it?
Okay, let's try that.
Sorry, everybody, for these technical difficulties. This happens sometimes with our virtual platforms. So just give us one moment and we'll get started.
Okay, so Jody at coloradotix.org, yeah.
Perfect. Yep. And then I'll go ahead and share it, and then you can just
Okay.
cue me when to switch those slides.
Okay. So I am sending it right now.
Perfect.
I'm wondering, does this have to do with speaker view? I'm going to try this again.
Okay. Yeah, you could try your views. That could change it.
That didn't change it. Hmm.
I've got it here, so I'm going to go ahead and share it. So let me get this opened. There we go. All righty.
There we are. And now can you go to the full screen? It should be bottom right.
There we go.
This is different. Aha. Okay.
Okay.
Go. And Jody, I'll just nod to you. You
Okay.
know, okay.
That'll work.
So here we are. Starting again, a toolkit for promoting your own healing. That's certainly my intention here. The next slide is just a cartoon which shows someone at TSA airport security with a whole bunch of bottles and extracts, et cetera. And the TSA person says, Lyme disease, huh? I'll just mention this. This is exactly what my wife goes through every time we travel. Okay, next slide. I want to talk about the seed and the soil, okay? The seed is the bug, the soil is our response to the bug. In chronic infection, you have to consider both, right? So first we're going to talk about the seed. What's the seed? Well, the germ theory, which is our first so-called scientific explanation of disease, really is only maybe 150 years old. And the I don't know, is it full on your screen right now, Jody? Just because it's not quite on mine, but maybe.
Yes, it is.
Aha, okay, fair enough. So, so basically in the late 1800s, we're looking at the mortality of women with childbed fever. These are women who gave birth and then became septic. And the mortality was really high when doctors delivered babies in the hospital, but it's important to note that before there were such a thing as hospitals and these women delivered at home with midwives helping them, the mortality was low. And then this Dr. Semmelweis, he watched what happened in terms of doctors going to do autopsies and then going to deliver babies and not even washing their hands. And he tracked what was happening. And he tried to convince his colleagues to wash their hands. He didn't know about germs. He just tried to convince them, wash your hands. And he couldn't convince them. He ended up going into an insane asylum and paradoxically, he died of sepsis. That's where we were. And then Louis Pasteur isolates bacteria from the blood of women with postnatal sepsis. You know, we had seen these bacteria. Van Leeuwenhoek back in the 16 or 1700s saw these little things. No one knew what they were. Well, Louis Pasteur made a connection and said, whoa, this is what's making people sick. And then Robert Koch, in pretty definitive test, he isolated the tuberculosis germ from people. He injected them into guinea pigs who then died of TB. Next slide. So basically, can we get the next slide? There we go. So that's the germ, you know, finally we have this explanation. Here's why people get sick. But even then, there were people who were saying, wait a minute, it's not just the germ. Robert Virchow, in particular, headed this line of thought, and he claimed it wasn't the germ that made people sick, it was the condition of the host. And representative of that is the fact that basically everyone in Europe in the 1800s had tuberculosis. Everyone was affected, but it was only those people who were stressed, had some impairment of their immunity, come in different shapes and forms. They're the ones whose activation became infected and developed active tuberculosis. Pasteur then rethinks what's happening. He goes to the, I guess it was the France medical convention in Paris, and he shows up with two birds, and he said, I injected both of these with anthrax. Bird number one was housed in a warm pen. He was alive, healthy, clucking. Bird number two, underfed, and exposed to cold, and quite dead. And this famous pronouncement from Louis Pasteur, La terrain is everything. Well, so he's jumped to the other side of the coin, but it's not everything. It's both the seed and the soil. Next slide. What we're talking about today is the soil. So I'm very grateful to Joe Birascano and all these others who've increased our knowledge and awareness of the seeds, but we don't talk that much about the soil. That's what we're going to talk about today. Okay, in the next slide, Joe mentioned this. You know, everybody's different. Sometimes I get asked, you know, how do you treat Lyme disease? And what I say is, I don't treat Lyme disease. I treat people. They're all different. And here's just some of the reasons how we differ in our genetics and biochemical individuality, epigenetics. These are changes in the genetics that can be transferred just from one generation to the next. Previous infections. Do you have indolent Epstein-Barr, for example? Diet, nutrition, obviously, exposure to mold and other allergens, exposure to toxins, mold toxins, heavy metals, other toxins. Lifestyle issues, stress, sleep, socialization, screen time, exercise, adverse childhood experiences. I will go into that in more detail. Adverse adult experiences. Doesn't help if, you know, you've suffered significant trauma in a car accident, for example. Okay, next slide. And so I want to talk about the mind-body connection. How do we understand it? Well, do factors impacting the mind impact the physical body? Yes. I mean, it took a while for Western medicine to figure this out, but it's now generally accepted that people who are stressed in any assortment of ways, it can affect us physically with all sorts of different illnesses. Does physical illness impact mental health? So this is the opposite direction. Well, it's generally accepted that if you're really sick, it can make you depressed or anxious. Okay, but we in the Lyme community know that these infections can impact your mental health directly via neuroinflammation. So then the question is, can we use the mind to influence our physiology, our response to these infections? Next slide. So what happens with stress? And when I say stress, this can be stress from an infection. It can be stress by being attacked by a mountain lion. It can be everything in between. Okay, in the next slide, I'm going to talk about, nope, that one.
There we go, sorry.
So stress stimulates the autonomic nervous system. When we talk about the autonomic nervous system, think automatic nervous system. You don't have to think about how fast your heart beats. You don't have to think about where the blood should go. You don't have to think about how fast to breathe or how deep to breathe. Basically, the autonomic nervous system takes care of it, which is good. If we had to remember to breathe, we'd be in trouble. Okay, so there's different arms of the immune system. There's the sympathetic arm, parasympathetic arm, and enteric arm. I just want to say I'm not going to be talking about the enteric arm. I'm going to be talking about the sympathetic and parasympathetic. So the sympathetic is mainly stimulatory. This is the accelerator, and it's most famous for when it hits the adrenal medulla, when that's stimulated, it releases epinephrine and norepinephrine, otherwise known as adrenaline and noradrenaline, and we get the fight or flight response. It also affects the hypothalamic pituitary adrenal axis, and we have increased cortisol under sympathetic stress. And this is really important. The sympathetic arm is pro-inflammatory. It increases inflammation. You're under stress, guess what? It's going to increase inflammation. The parasympathetic arm can oppose the sympathetic arm. So we have now a brake. The whole point is that if we have an accelerator and we have a brake, the point of the autonomic nervous system is to keep us in homeostasis. So the parasympathetic arm is relaxing or calming, and it is anti-inflammatory. Okay, next slide. So I'm not going to go through the slide. I just want to point out that the autonomic nervous system, we're hitting all the organs, everything in the body. You know, we're only going to be talking about a little piece of it, but it's everything. Everything is regulated to some degree by the autonomic nervous system. Okay, next slide. So the vagus nerve. The vagus nerve is the 10th cranial nerve. The cranial nerve comes off of cranial nerves, in all of them, they come off the brain, whereas other nerves come off the spinal cord. The vagus nerve is the 10th, and it is the main contributor to the parasympathetic nervous system. It is afferent, which means it actually delivers feedback from the heart, lungs, and gut to the brain, but it is also efferent, which is that it's stimulating the body. And in that sense, opposes the sympathetic nervous system. As I mentioned before, it's calming. So when the parasympathetic nervous system gets involved, it lowers heart rate, lowers blood pressure, dilates blood vessels, dilates bronchioles to breathe deeper. It's calming, and it is anti-inflammatory. We'll talk about that a little more as well. The obvious take home here is parasympathetic is good. Okay, next. However, there's two branches of the vagus nerve, and this comes under the headline of polyvagal theory. So it turns out that what we associate with parasympathetic activity really has to do with the ventral vagal arm or branch of the vagus nerve. So this is the parasympathetic calming I've been talking about, whereas sympathetic is sometimes referred to in its innervation with causing fight or flight. Sometimes this is referred to as tend and befriend. By tend, I mean you take care of yourself, you take care of your kids, and if there's a threat, you try to befriend them rather than fight or flee. Okay, so what does that mean? It means social engagement, connection with face and heart, which, you know, because these are expressions of engaging with people around us. However, the dorsal vagal complex has a very different effect. It leads to freeze or paralysis. This is suddenly people, you know, deer in headlights, okay? When people experience this, they can't react well. They go into deep depression. They feel disconnected. They feel hopeless. It's surrender. They want to hide under the covers and not come out. This is the dorsal vagus, okay? So next slide. Here we have, I'm going to show you a series of slides. There'll be huge redundancy, but I'm really trying to drive this home. So you can see that the ventral vagus nerve is, you know, basically encourages social engagement, whereas the dorsal vagus immobility, and then there's the fight and flight, the sympathetic nervous system. So that's the three-part autonomic nervous system. Next slide. And here you see, you know, you can also see this sort of from an evolutionary standpoint. On the left, we see immobilization, we freeze. And, you know, this is what reptiles do. This is a very low on our evolutionary tree that in terms of reactivity. The next level would be, whoa, there's danger, let's get out of the way. But further up on the evolutionary tree is our ability to feel calm and connected and engage socially. Next. And here it is again. I mentioned that I'm repeating myself, but it's the same things, you know, start at the bottom, immobilization, despair, hopeless, collapse. Whereas sympathetic, stress, anxiety, fight, flight, conflict. And then there's the ventral vagus, social engagement, feeling safe, calm, regulated, balanced, resourceful, resourced. Okay, next. And so we just went through past, you're gonna have to go backwards again. This is one more slide. And it just basically, you know, each slide was to give you just a few more details of what this looks like, you know, with the freeze, dissociation, numbness, depression, helpless, shame, shutdown, hopeless, wanting to die, feeling trapped, and then sympathetic, rage, anger, irritation, frustration, panic, fear, etc. And then social engagement, feeling grounded, compassion, curious, open, connected. Okay. I promise not to repeat it anymore. Next slide. Now I want to talk about trauma. What's trauma? Well, trauma I'm calling stress on steroids. It's not just, oh boy, I have an exam tomorrow. Trauma is big time stress. Next slide. So trauma, no surprise, dysregulates the psychoneuroimmune endocrine system. So believe it or not, I did not make that up. Those who've been around for a while are aware of the term psychoneuroimmunology because it's very clear that what we feel from a mood perspective affects our nervous system and immune system, and they're really not disconnected, but it's been expanded. It affects our endocrine system too. And guess what? They're not separate systems at all. This is all part of one big interconnected function in our bodies. So when trauma dysregulates this system, we see dysregulation of the autonomic nervous system, what we've just been talking about. And these are common sequelae of that: mood instability, hypervigilance, is it safe? Inflammation, remember, pro-inflammatory, immune suppression, and post-traumatic stress disease. This is a quote from Bessel van der Kolk: trauma radically changes people that in fact they are no longer themselves, or at least who they thought to be. So on the next slide, I'm going to talk a little bit about Bessel van der Kolk. So Dr. van der Kolk is a psychiatrist. He was at Harvard back when I was in Massachusetts practicing then. Coincidentally, I knew his wife and kids, but I've never met him. And he has written an enormous amount and has taught an enormous amount about the effects of trauma, particularly childhood trauma. Now, what was very interesting to me is that at the time in my medical practice, I treated a lot of people with environmental illness. We had a large clinic, you know, we had full spectrum allergy testing, et cetera. And it became clear to me that there was a certain connection between those who had the most severe environmental illness and a childhood history of trauma. So again, severe environmental illness, these are people who were reduced to eating only a handful of foods, who had severe multiple chemical sensitivities, could not go into public places because a whiff of perfume was enough to give them migraine for a day. And talking with these people suffering from this level of sensitivities, a preponderance of them at that time had histories of childhood abuse. And interesting, while I hadn't met Dr. van der Kolk, I heard lectures he gave in which he said, you know, these people with trauma, that childhood trauma, they develop a lot of physical problems. We were seeing, in essence, the same people coming at it from the two different directions, he from the psychiatric and myself from the organic. But basically, it was all connected. And here's how we connect it, that trauma leading to, boundary issues, what's safe, what's not safe, leads to hypervigilance. Just, you can imagine that someone who grew up in an environment that wasn't safe is always looking out for what's going to happen next, who's coming through the door, etc. Hypervigilance in the nervous system translates to hypervigilance in the in our immune systems. Remember, these are really not separate systems. It's all interconnected. So what happens when our immune systems are hypervigilant? More inflammation, multiple sensitivities, and environmental illness. I wrote about this back then, and I had other practitioners contacting me saying they were seeing the same connection between severe environmental illness and significant childhood trauma. So Bessel van der Kolk wrote a book called Your Body Keeps the Score. It is a fantastic book. I can't recommend it enough if you're interested in reading about trauma, childhood trauma in particular, but not only childhood trauma, and post-traumatic stress disorder. Okay, so the next slide, we're going to talk about PTSD. So the concept of PTSD was first developed to describe a syndrome soldiers experience during war-related trauma. Back in World War I, this was described, it was called shell shock. There are genetic and epigenetic risk factors to PTSD. Something occurs, and it undermines a basic sense of, am I safe? Am I secure? And PTSD results in dysregulation of the limbic system, which is our emotional brain. It goes a bit haywire. Okay, next slide. Okay, what does it do exactly? PTSD causes hyperactivation of some brain structures while others become hypoactive. So the hippocampus, which stores memories, it shrinks and it distorts the memory, including causing dissociative flashbacks. The amygdala becomes overly reactive. Amygdala is sometimes referred to as the heart of the limbic system, which is the emotional brain, and that leads to this autonomic response. It also releases norepinephrine in the brain, which causes tremendous anxiety. Meanwhile, the medial prefrontal cortex, that's the gray matter, which, remember, it evolved later than the limbic system. It takes longer for information to get there, and it has inhibitory control over the amygdala. But with PTSD, it becomes smaller and less reactive, and it fails to modulate a response to perceived threat. So think about that. If you're reasonably calm, cool, collected, and someone does something threatening, and you have good resources, you don't suffer from PTSD, the prefrontal cortex will say, okay, what's my best way of dealing with this? Versus someone with a significant history of trauma who may very well freak out. Okay, next slide. So this is from Dr. Robert Bransfield. Joe made reference to Dr. Bransfield. He's written more about neuropsychiatric Lyme than anybody. He's fantastic. And this is from a talk that he gave. And I just want to point out that if we go to the left and see an external danger, that it results in fear, appropriate fear, response circuits, what do we do to deal with this threat? And we're on the wrong slide, there we are. And if you're well-resourced and you're able to deal with the threat, you adapt, you're good. You don't get PTSD. But let's say you don't have an ability to respond to this threat. It's something out of your control that can lead to PTSD. Well, PTSD can lead to inflammation and autoimmunity. I already talked about the physiology of PTSD and how it affects our nervous system and so on. We'll talk about that more. So all that leads to multiple psychiatric and somatic symptoms. Sound familiar? Well, it's the same thing that happens. Look at the right-hand side of this. Internal danger, which would be like tick-borne infections, which We need a good immune system to deal with it. But let's say it's overwhelming infection and we don't get proper treatment for it. Well, the same thing. It goes to persistent inflammation, autoimmunity, and guess what? That in itself can then trigger the limbic system, PTSD, and we got a snowball going here. On the other hand, if we do get appropriate diagnosis and treatment when we get infected, we can adapt and boom, we can stay healthy. Okay, next slide. So here it is again, PTSD dysregulates the psychoneuroimmune endocrine system. Endocrine is bolded because that's what this slide is about. Basically, distortions in the hypothalamic pituitary adrenal axis, the autonomic nervous system, hormone dysregulation. So we see not only release of epinephrine and norepinephrine, fight or flight, heart rate, blood pressure, arousal, startle response, but abnormal cortisol regulation. So now we have immune dysregulation and even resistance to receptors to cortisol, leading to increased inflammation. Next slide. And now we're going to talk about effects on the nervous system, but you know, you can see how you really can't separate these out. This is all happening simultaneously. So what do we see in the nervous system? Well, there's an increase in glutamate. This is in the brain, which fosters dissociation and derealization. You know, think of what happens to people whose dorsal vagus is stimulated and they go into that paralysis place. An increase in brain norepinephrine, I mentioned this before, generates fear and anxiety. A decrease in GABA activity. Remember, GABA is an inhibitory neuropeptide. It is normally calming, but there's a decrease in that activity. And as I've already mentioned, activation of the autonomic nervous system. Next slide. And here's what happens with immune dysregulation. So PTSD results in a hyperinflammatory state. There is an increase in pro-inflammatory cytokines, a decrease in anti-inflammatory cytokines, and an increase in autoimmunity. So there's a suppression of immune function. And as I mentioned previously, hyperinflammatory responses and hypervigilance lead to increased sensitivities. And then summing it up, hyperinflammatory states lead to multiple somatic and psychiatric symptoms that may further trigger PTSD. Okay, so now we have this cycle of inflammation and symptoms. Next slide. So, you know, this is a summary. What we see is dysregulation of the autonomic nervous system, a lot of mood instability, hypervigilance, inflammation, multiple sensitivities, immune suppression, hormone imbalances. Basically, PTSD impacts our sense of self in the world, of who we are. Like, am I safe? Am I loved? Is the world safe, etc. Okay, next slide. So childhood trauma is PTSD on steroids. So PTSD is a big category, can manifest many different ways. And I just want to point out that when terrible things happen to an adult, like say a war vet from Vietnam or Iraq or Afghanistan, or a rape victim, God forbid, or you know, someone in an absolutely terrible car accident, these can easily generate PTSD. Childhood trauma is different. And here's what's different. Those people who suffered it as an adult, hopefully they had a reasonable enough childhood that they have some resilience and they have some resources and they have a sense of who they are, that they developed a strong sense of self and safety in the world and what they can do. When one experiences significant trauma as a child, it's, you know, who knows what's safe? People are supposed to be safe or not safe. What's a healthy boundary when people who are supposed to love you unconditionally instead can be violent and so on? I mean, you get the idea. So in the next slide, this is started off with a famous study. It was done at Kaiser about adverse childhood experiences, which generates the so-called ACE scores, okay? So what they did was they actually looked at 17,000 people, adults, and they asked them to fill out a questionnaire. Did they have any of these adverse experiences that are listed here? And then they compared their medical charts in terms of their physical illnesses. Okay, it's a given that people with significant ACE scores are going to have much higher preponderance of psychiatric issues, whether it's anxiety, depression, or suicidality for that matter. But we're looking at physical problems now, okay? I'm just going to read these for the record. Psychological abuse, physical abuse, sexual abuse, emotional and or physical neglect, loss of a parent for any reason, a mother treated violently, substance abuse, mental illness, criminal behavior in the household. So these are things that happened in your family of origin. Okay, in the next slide, I just want to show you something. This is really amazing. So if someone had an ACE score of 2, just 2, the increased risk of hospitalization for autoimmune diseases, it's an increase of 70 to 80%. Now, that doesn't mean you have a 70 to 80% chance of being hospitalized for an autoimmune disease. It means you have an increase of 70, 80%. In other words, it goes up from 1% to say 1.75%. That's a huge increase. And for every increase in the ACE score of 1 point, from 3 to 4 to 5, the risk of developing an autoimmune disease, such as type 1 diabetes, lupus, rheumatoid arthritis, and many others, each step it goes up 20%. And I need to remind you, Lyme disease complex is an autoimmune illness. Joe just gave a wonderful talk on Bartonella, which causes more autoimmunity than any microbe that I've experienced. Okay, next slide. And just to point out, ACE score and risk for other chronic illnesses that they've looked at. That top line, it's now this is with a score of 4 or more. There's a 4 times higher risk of lung disease, 2.2 times higher risk of heart disease, almost 2% higher risk, not 2%, 2 times higher risk of cancer, 1.6 times higher risk of diabetes. You see my point, you know, trauma affects our physical bodies, childhood trauma even more so. Next slide. And PTSD is not only in victims of obvious trauma. So think about, maybe these have happened to you, chronic pain symptoms that cannot be controlled, chronic illness without explanation, chronic neuropsychiatric and somatic symptoms that make it hard to cope, negative encounters with physicians who are arrogant, ignorant, contemptuous, and dismissive, neglect and isolation, chronic inflammation and autoimmunity. Sound familiar? I think all of us who've experienced Lyme disease complex are acquainted with these kinds of traumas. Next slide. So let's put it together. Lyme disease complex and PTSD. I'll just remind you that I use the term Lyme disease complex. I explain this in my book because once we're talking about chronic Lyme, it's not just Lyme. As Joe pointed out, we're talking about Lyme plus co-infections. We're also talking about all this dysregulation in the nervous, immune, endocrine system, and so on. So what do we know? Lyme itself can be traumatic and cause PTSD for all those reasons in the previous slide. Lyme disease complex and PTSD result in the same overlapping pathophysiology. What do we see in Lyme disease complex? The same dysregulation that I described in people who suffer PTSD in the nervous, endocrine, and immune systems. And Lyme disease complex can directly result in activation of the limbic system. And there it is. There's your PTSD. Next slide. So Lyme disease and complex and PTSD are all the result of trauma, leading it to changes in who you believe yourself to be, like Bessel van der Kolk described. For example, do you think of yourself as, I'm a worthy person, I deserve good things, I'm lovable, people like me, I feel secure and safe, or not? Do you feel like you're worthless? Do you feel like there's nothing about you that is lovable? Do you think you're not a good person? Do you feel like there's nothing about you that is safe? And then what about the outside world? How do you perceive the universe? Is the outside world safe, or is it in fact hostile or dangerous? Is it supportive or basically not caring? I mean, we could go down a whole list. So what I want to point out is that PTSD pre-existing Lyme disease complex will make symptoms and treatment more challenging. Okay, next slide. And here we have Dr. Bransfield's slide again. And I'm just reminding you how, you know, the external danger might be what happened when people grew up and had childhood trauma, but it can also be, you know, adult trauma and the fear response circuits that leading to PTSD symptoms, and then combine that with tick-borne infections, immune system problems with inflammation, autoimmunity, and wow, you know, it's a perfect storm. Okay, next slide. So here's a story. What happens if you get attacked by a mountain lion? Those of us in Colorado probably all know this story. This happened, I think, around 2019. There was this dude, he was probably around 30-something years old, and he's basically jogging or running outside of Fort Collins, Colorado. He hears this clip-clop behind him, and suddenly he's attacked by a mountain lion. Yeah, these things don't happen too often, but here's what happened. So first he had to get the claws off his face. And then, you know, there's an interview with him, and excuse me, so I went ahead and wrote a link here to the interview. There's an interview with him that I watched, and, you know, he's calm, cool, collected, you know, but he's not bragging or anything. He's just being normal. He said, well, what happened was, I remembered that, you know, I had cats when I was a kid, and we used to play with the cats, and when they were on their backs, they were helpless. They really couldn't use their bodies to do anything. So after getting the claws off my face, I wrestled this mountain lion onto its back, and then I strangled it to death. Wow, that's, believe it or not, that's what he did. And in the next slide, you can see a picture of this dude who's got a whole bunch of stitches on his face, but he's actually none the worse for wear. And what I'm pointing out here is this dude was able to deal with the event. Sure, he had an autonomic response, an appropriate one, saying, oh my God, I'm in danger. And then he was able, thank God, to deal with it. He's not going to have PTSD. Not only that, but I can guarantee that this person had a good enough childhood that he did not grow up as a victim, that he did not grow up as helpless, but rather empowered. That he has a sense of self as safe and secure and able to handle things. Thank God. Okay, next slide. So here it is. You know, again, this dude from Fort Collins on the left, external danger, fear response circuits, and then adapts to the danger. No PTSD. He's good. Next slide. So how do we heal? Okay, this is not easy. I think we can all agree on that. And I'm going to break it down. So in the next slide, Jody, there we go. I want to talk about the mind-body universe. So the mind, I'm referring to the mind not as our thoughts. This is what I'm thinking right now. It's our belief systems about ourselves. I made reference to this before. Who do we believe ourselves to be, and how do we believe the world will be in response to that? Okay, you know, are we inherently safe and secure, or do we feel vulnerable and in danger? Do we see the world as hostile or caring and supportive? Okay, the mind actually, it holds a template. Our belief systems hold a template for our emotional body, which is also the world of energy, the world of feelings. And that in turn holds the template for the physical body, the world of action. All these worlds are all here right now. The next slide. Jody, the next slide. There we go. No, we missed a slide, I think. We missed a bunch of slides. So back it up. Okay, try again. Okay, so what can we do about this? Well, let's start off with the world of physicality and doing, well, you've heard all about these things. I just listed them here. We need to acknowledge the problem. We got to make a diagnosis, right? Excuse me. And I want to point out that in some patients, in some patients, it's medications to help stabilize our nervous system and our mood issues are important. I can tell you when I had severe anxiety with panic attacks, severe depression in which I thought about suicide on a daily basis, medications were important, okay? This was not a time when I could deal with, say, childhood trauma or relationship issues. This was basically, I just needed some sort of stabilization to get through the day and not want to kill myself, okay? So it's very important. It's very important in that stage to use medications and or supplements for calming, mood stabilization, pain, and sleep. And I'll just point out that sometimes the best thing we can do then is also just plain old distraction, you know, binging on TV, if that's what it takes, or, you know, reading books, whatever. You know, I think I read the Fellowship of the Ring trilogy twice. Okay. What else? Decrease ongoing negative stimuli, environmental triggers, which include foods, chemicals, and mold, avoiding toxic exposures, toxic people, toxic relationships, avoid the news if it's upsetting. And we all know there's pretty upsetting news out there. These are all things you know: diet, exercise, sunlight, antimicrobials, of course, as tolerated, balancing hormones, supporting the immune system, detoxification. Joe went through these as well. These are things you know about. This is actually not what we're talking about today. In the next slide, we're going to talk about the world of emotions and energy. And basically, this is really about incorporating activities to enhance the ventral branch of the parasympathetic nervous system. Excuse me. I don't know if this is totally inclusive, but it was everything I could think of: meditation, relaxation, deep breathing, mindfulness, yoga, qigong, tai chi, body work like massage and craniosacral work, biofeedback, acupuncture, body work, EMDR, polyvagal exercises. These are 2 books on polyvagal exercise, Anchored by Deb Dana and Accessing the Healing Power of the Vagus Nerve by Steve Rosenberg. Tapping on acupressure points. It's called Emotional Freedom Technique. It has other names like Thought Field Therapy and Callahan Technique. Music, chanting, dance, rituals, equine therapy. The point is there's no one avenue to take. We have a potpourri, and some things work for some people, some things work for others. Okay, support is important for everyone. You don't do this by yourself. You need to be connected. So whether that's groups or just individuals that you're talking with are important. Talk psychotherapy can be very important to get that kind of support. And body-centered psychotherapy, which is to help people really feel into themselves and also to help move trauma out of the body. And finally, MDMA, you know, that's ecstasy, right? Now there are studies, and in fact, one of them I think is I think the primary investigators in Boulder or Longmont. It's anticipated that MDMA will be approved by the FDA next year for the treatment of PTSD. It has had a very high success rate. And what we're talking about, this is not a hallucinogen. This is a modality that helps people in a very safe space with a very good therapist guide, relive their trauma in a non-traumatic way, and eventually being able to say, you know, that happened then, but this is now, and not be feeling like that trauma was still happening in their bodies. It's putting it in the past and then feeling the present. In the next slide, we talk about the mind. Okay, again, this is the world of belief systems. And here we're trying to actually desensitize the limbic system, which is so trigger happy and just goes nuts when something happens that somehow feels or sounds or smells like some trauma. Okay, so there's two programs, DNRS, Dynamic Neural Retraining System, and the Gupta protocol that seek to do this. I'll come back to describe them. Excuse me, they basically have to do with imprinting, you know, positivity instead of the negative trauma. I need to point out that in order to do these programs, you need access to childhood scissors or to trauma scissors. That is, if those feelings come up of that terrible car accident, the terrible rape, or the terrible childhood trauma, you need to be able to say, no, I'm not going there. You need to be able to edit it. You need to be able to switch the channels from a bad memory to a good one, even if you make it up. The point is that people who have a really strong PTSD, they're not ready to do this work. This is just on another level, because in order for this to work, you really need to be able to cut off those negative feelings. In the next slide, there are other ways that this works. Plant medicine. So psilocybin, LSD, these are hallucinogens. There are a lot of studies, mainly with psilocybin. So the reason psilocybin is studied more than LSD is because a typical psilocybin journey is 4 or 5 hours, and LSD is like 6 to 8 hours. So if you're going to do a study, you want to do something that doesn't take that long a time. But we're talking about, again, guided journeys that help, in this case, you know, really help see them people in the world all by themselves. You know, this is interesting. I'll just mention that when I was a medical student, which goes back to the early 70s, in neuroscience class, they showed us a video of an individual giving LSD to people with terminal cancer and sitting with them during a journey, being a guide, holding their hands. And when they came through the other end, these people were no longer afraid of dying. It felt like they had gone through a death process, and they felt now, you know, there was nothing to be afraid of, and their hearts were wide open. What was being done then is now being reproduced now in major medical centers like NYU and Johns Hopkins. Other studies using these modalities have worked with depression, they've worked with addiction disorders, and so on. I highly recommend Michael Pollan's book, How to Change Your Mind. He's an excellent writer, and he does great research, and he describes how these things work. I want to point out that healing on this level of belief systems will not hold unless healing work is also done on the emotional and physical levels. Some people feel like, oh, you know, if I can change how I believe, then that's going to affect my emotional body, my physical body. And temporarily, that might be true, but it won't hold. So really, folks, we have to work on all these levels. We can't do the so-called spiritual bypassing. Okay, so in the next slide, I'll talk about dynamic neural retraining system, really just to say, here's where you can access and learn about it. This is a quote from their website. And basically what she's saying is that it creates structural and functional changes in parts of the brain. We're talking about stabilizing the limbic system, and they're doing research in people on that. In the next slide, I talk about the GUPTA program, and he's really doing the same thing. Both these programs take a big commitment. And by that, I mean, they ask you to be doing some work on a daily basis. And so Gupta, Ashok Gupta, he refers to what he does as amygdala and insula retraining, or AIR techniques. Again, he's really desensitizing the amygdala. And he says that they have a clinical audit. We found that 2/3 of their clients reached an 80 to 100% recovery within a year. I know people who've done this, they've had great success. I also know people who've done it for just a month or two and it didn't work. It really takes at least a 6-month commitment. Okay, next slide. Story number 3. Okay, this is my last story, but I think it's a great one. So there's a town in Pennsylvania. It's actually not that far north of where I grew up, which was Allentown, Pennsylvania. Rosetta, Pennsylvania is north of Allentown. It's sort of very close to the Delaware River. And back in the 1960s, actually in 1960, it drew the attention of some epidemiologists and a social worker person from the University of Pennsylvania. And it drew their attention because the incidence of heart disease, which then and now is the most common cause of mortality in the United States, was half the national average. So they went into this town wondering, like, well, what's the story? Are there a whole lot of, vegetarian marathon runners or something like that? Well, back then, these towns could be a predominance of one ethnic group or another. So Rosetta, they were mainly Italians. Other towns might be Welsh or Irish, excuse me. But these people actually were progeny of families who came from Rosetta, Italy, somewhere south of Rome. And when the quarries basically closed up there, they needed to move, and they started coming to the US, and some families came together, and they actually even brought their priest over. So there was this incredibly tight community where people did things together. They all went to the same church. They had tons of meals together. There was like multiple parents to the same kids because there was so much closeness in the community. And, this is a quote from a review on Rosetta, Pennsylvania, and I'm going to read it. Despite a greater prevalence of obesity in Rosetta, yeah, these people are just eating pizza and hanging out on their porches, and despite similar dietary, smoking, and exercise habits and similar ethnic and genetic background, the inhabitants of Rosetta were relatively immune to heart disease at the beginning of the research in 1963. By the way, they were also immune to a lot of other health problems. They were also strikingly tenacious in adhering to old world values and customs. When these traditional values and relationships were abandoned by the rising generation, death rate from heart disease climbed toward the American norm. The investigators that went in there said in 20 years, the rate of heart disease was going to be the same, because they could see what was happening with the next generation who, guess what? They didn't really want to go to church. They didn't want to hang out with their family. They wanted to watch TV and hang out together. They were doing what American kids were doing. The study concluded that unconditional interpersonal support counteracts life stress and thus preserves life. I've just always been moved by what happened in Rosetta, Pennsylvania. And a short personal story is, I would read about this over and over. And one time I said to myself, what was it like? What was it like to live there? And I actually, had this sort of internal vision of myself. I'm standing on a porch of this brick row house. I'm looking out over this town, and I feel like, here's what I felt like, that, oh, I'm just sort of an average bloke. I mean, there's nothing special about me. Some things are pretty good, some things are not so good, but there was an incredible sense of belonging. There was belonging and acceptance I was a part of this, and there was an unconditionality with it. And I can tell you that the overwhelming sensation in my body was of this deep relaxation and safety. It was amazing. This experience that I had, deep relaxation and safety in community. Okay, next slide. So this is a summary slide. What's it about? Well, the mind and body are aspects of a single universe. And I say manifesting on different vibrational levels. I don't know how else to say it. Vibration doesn't quite make it. The point is, it's all here now, and it's all interconnected. Stabilization of the psychoneuroimmune endocrine system leads to emotional and physical resilience. And this is a quote: "The roots of resilience are to be found in the sense of being understood by and existing in the mind and heart of a loving, attuned, and self-possessed other." In other words, healing occurs in relationship. You don't do this by yourself. We need others to hold us in that place of unconditionality. Healing has occurred when an individual experiences safety, real deep internal safety with healthy boundaries. That's it. Thank you very much. And the last slide, I just want to thank Robert Bransfield, because I used some of his slides in this presentation. Thank you very much for your attention. I'm happy to take a few questions.
Yeah, so far, we don't have any questions that have come in.
Wonderful.
Does anybody, kind of a last minute, does anybody have any questions before we wrap up today? Okay.
I actually see a couple here.
Oh, I don't. That's weird.
Yeah, here they're coming in.
Let me scroll. I'll scroll down
Yeah,
here.
scroll down, yeah. There was one from Tara. While you're scrolling, I'll go ahead and take this. This is from Tara. She's saying, feels so on point to me. Thoughts on complex PTSD versus complex PSD. Whoops. Other questions are coming in. Sorry, just moved that on me. Thoughts on complex PTSD versus PTSD. My therapist doesn't believe in complex PTSD as it's not in the DSM book, but she's also not so knowledgeable on tick-borne infections.
And I'm sorry, what was the question?
What your thoughts were between complex PTSD versus PTSD?
Okay, I actually am not aware of that nomenclature of complex PTSD. But I just want to repeat what I mentioned about the book by Bessel van der Kolk, Your Body, I got it right here, The Body Keeps the Score. It is an outstanding book. I'm jealous as an author, because he wrote such a fluid book with so much information that is easy to read. It flows. He's got a wealth of knowledge and experience, and then he draws from the literature and from his colleagues, and he puts it all together. So I just recommend that to anyone who's dealing with these issues.
This one is from Amy. What do you do to decrease herx?
Oh, okay. Well, again, I talk about this in my book. It's sort of herx is really inflammation associated with killing a microbe. Some people say, well, when I detox, I feel a herx reaction. That might be an inflammatory reaction, but it's not a herxheimer, which by definition it has to do with taking an antimicrobial. There are a whole lot of things I do, which include perhaps cutting back on the antimicrobial agent. There's alkalinization. I suggest water with a lot of fresh lemon squeezed into it, trisalts or Alka-Seltzer Gold, binders like cholestyramine and charcoal, baths with a lot of Epsom salts, and baking soda, as well as there's some herbs like Berber Pinella from Nutramedix, which can be very helpful. So, and different things work in different people. You know, one point I want to make is that Herxheimer's are not good for you. If someone has acute Lyme disease, then and they were otherwise or healthy, but we're seeing them and they went from being healthy 1 month ago and now they have acute Lyme disease. They'll tolerate a Herx, they'll go to the other side and be just fine. But we need an infrastructure before we can tolerate Herxheimer reactions. So most of my patients with chronic Lyme disease, there's some degree of fragility there. And if they have a severe Herxheimer reaction, it's going to reset them to a point where they're more inflamed and feel worse and take a long time to recover. So I just want to point out, it's not a good idea to push these high suffering Herxheimer reactions if one has the chronic infections.
So we have a question from Nanette. Are you taking new patients?
Sadly, I am not taking new patients, no.
Okay, we have a question from Francis. Can you talk about somatic therapy and how it works with PTSD?
Yeah, well, here's the thing. Again, I recommend people to Bessel van der Kolk's book, but what we're talking about is trauma resides in the body. It's not just your head. The trauma can be located by people who can, if you ask them to tune in, they'll say, oh, this is where it hurts. And so somatic-based therapy or body-centered psychotherapy is helping people access the trauma, the feelings associated with it, and allowing it to move. The important part being, this needs to be done in an atraumatic way. It needs to be done very gently. Peter Levine, who is actually right here in Boulder, who's nationally known therapist dealing with PTSD and trauma, he talks about just dipping your toe in and then taking it out, dipping your toe in and taking it out, and you know, slowly getting people tolerant of visiting what happened to them, and eventually getting to the point where people can describe it. This is what happened. Actually articulate, here's what I felt, here's what happened. And then, you know, get to the point where this happened in the past, it's not happening anymore. That's what the therapy is. That's what it says.
Oh, I'm sorry.
Yeah.
Mara, how do we experience healing community when we are in physical isolation?
Wow, that's a great question. You know, even before COVID, our population was increasingly isolated. The numbers are terrible. We need to seek it out, and it's going to be incredibly imperfect. You know, I just make it a point. I talk with my best friend in Massachusetts every week. I talk with my childhood friend from nursery school on a regular basis. I try to go out to lunch with my friends, just connection, and any way we can. And unfortunately now it's going to a lot be by Zoom, perhaps. You know, COVID has been a disaster, you know, in terms of our capacity to connect.
Okay, I don't see, Jody, I don't see any other questions. Are there any others? Yeah, I don't see anything else at this point. Okay, if anybody doesn't have any questions, thank you so much, Dr. Kindelier, for joining us today in your presentation. It was wonderful.
Thank you. Thank you. And thank you for making all this happen. I really appreciate everything you do at the Colorado Tick-Borne Awareness Association. It's great.
Thank you.
Thank you. Thank you all for joining us today. And I will be reaching out to those lucky winners for the door prizes from Intech Shield. So again, thank you everybody, and enjoy the rest of your weekend, what is left of it, and have a fantastic week.